Thank you. Well, welcome everyone. Thank you for joining me today here. Let's see. So I have a fancy title and a not so fancy title. The fancy title kind of captures the science that I do. And the not so fancy title is, how did I get to where I am right now? So what are sort of, what's the path that I followed to get to where I am? So the subtitle, alternate title would be Career Options in the Interdisciplinary Sciences, because it is a very interdisciplinary science that I perform. And probably most of the slides you'll get that sense. So the first portion of my talk will be an introduction of myself. Then I'll follow with an introduction to my science. And then I'll give you an overview of some of the science I do. So it won't be super intense to start. Hopefully it'll be, fairly. I'll ramp up the complexity of my talk, so you won't get lost, right off the bat. So this is a slide that I've been working on for several years. I just added a few things to it because information is still correct. So I was born here in the U.S., in New York State, but I actually grew up abroad. I grew up in Netherlands. So I went to high school in Netherlands or Holland, and then I came back. I came back to the U.S. for college. In high school, I had a science-heavy education, and I continued that trend in college. I initially decided to do some bio, and then I decided on chemistry. Then I decided, maybe biochem is a good, mix between those. So I declared my biochemistry major in about 2 years in, and I did some internships in that field, bioinformatics, involving a little bit more computer work. I had done some lab work, and I really liked biochemistry, but I also liked the computer science part of that research. So I ended up doing two majors, biochemistry and computer science. And even though it took me 5 years to graduate, I think that was a good decision. In a sense, those were two things that I was good at and I liked, so I decided I'll do both. Now, I graduated and I went to grad school. I did my degree in biomedical engineering, so it's a more medical field. Even now, most of what I will talk about will not involve that degree that much. But I did my Ph.D. in biomedical engineering. I did some pre-thesis work on computational biology and some structural modeling. And then I eventually did my thesis in bone disease, osteoporosis. And later on, I did a postdoc and got a job, etc. But that's probably not the portion you're very interested in at this point. So what I'm going to focus on in the next few slides is this portion over here in the red and transitioning from the red into the blue, because I suspect that might be where you are. So I always like to put a map, because I'm a visual learner. So this is Google Maps showing New York, most of New York. So I own a college. That's where I went to undergrad. Then I went to grad school, Stony Brook University, which is a state university of New York. And it's roughly located in that Brookhaven National Laboratory circle right there. That's where I did my thesis work. And then for my postdoc, I went to Cornell. So very much New York State. Just so happened that way. First I'm doing this little word cloud thing. But if I look back to my 5 years of college, and I try to remember what were topics that really kept me busy, kept me excited, I think this captures it. This is the reason I went to grad school. This is the reason I went into science. Right? Things like chemistry and research really, really got me. The idea of doing experiments or how to do them better. I liked computers and computing. I was involved in some research projects outside of class. So this is my personal word cloud. Kind of, peek into my brain, so to say. So it's kind of cool to do this, actually. Kind of write down a bunch of words, and then you can pop this into one of those generators and see what you get. Now, during undergrad, as all of you know, you're taking classes, mostly in your major. There might be some electives, and if you're first or second year, that's your focus. What major am I going to declare? How do I do well in class? What do I want to take next semester? But as you get to your third year into your fourth year, maybe you're thinking about what's next. Am I going to go get a job? Do I want to go to grad school? Do I want to change majors, maybe, right? So for me, this involved, how do I get into grad school? Because I knew I wanted to get in grad school, but I didn't really know how to do that. I didn't have a lot of guidance in that sense, like you have. I think you have a lot of guidance. Things like this current topics in science, I didn't have that. And I think this is awesome. You get to see how other people did things. I really didn't have that, so I had to kind of figure it out and seek out those resources. So aside from what you probably know and heard from people, here are my 2 points of advice for you if you are looking to go into grad school. First of all, and it might sound trivial, you have to figure out who are you? Are you someone that likes science, that likes chemistry or biology? It might be obvious, but it's good to identify the things you like to do, you're good at, and kind of give yourself a 1 minute or a 30-second summary of, like, here's who I am. If I have to introduce myself to someone, what do I tell them? So if you can do that in 30 seconds or a minute, I think it helps define yourself, at least who you are right now. That's always changing. And the second question is, it can be very hard too, is if you want to go to grad school, then why? Why do you want to go to grad school? What is graduate school going to do for you? Maybe you have a certain job in mind. Will grad school get you there? Maybe you don't need grad school. If you're out to seek more knowledge, or if you want more training, is grad school the right approach? So you have to figure out, why specifically are you going to go to grad school? And what's grad school, whether it's 2 years to do a master's degree or a Ph.D., what is that going to add to yourself? So for me, answering these 2 questions helped me in that path getting into grad school. Now, while I was in grad school, things changed. Because at this point, great, I'm in grad school, I did my entrance exam, I got in, now what? So in grad school, obviously, there's a lot of studying, you're taking classes. So becoming a subject matter expert is a big deal, right? That's one of the goals. It's pretty obvious. You're going to take a lot of classes in your major. But more than that, I think grad school is about being a critical thinker. That's something you cannot get out of a textbook. That's something you have to develop. And that can be really hard. How do you take a piece of knowledge and to really think critically about that and form some original thoughts? That is essentially what I think what getting a Ph.D. is about. You have the collective pool of knowledge, and you're trying to add something new to that. trying to figure out what is known, what is not known, and you're trying to answer a question that no one's thought of and no one's answered. So those are your original thoughts. So if you can do that, you get a PhD, basically. Yeah, that's what it comes down to. So very simple. It just takes a lot of work, and it takes a lot of time, and I think that's if I had to put it in 3 bullet points, that's what it boils down to. Now, very briefly, just to finish my profile, this is what I did in grad school. My PhD was in biomedical engineering, and as I mentioned, I did my research in bone disease or osteoporosis. And usually, when you pick a research topic, you define the research topic. You figure out what is something that's worth researching. Maybe there is a big problem out there, and I want to dedicate my career to helping solve that problem. And for me, that was osteoporosis. Osteoporosis is a bone disease, and there's all kinds of facts I could put on the board. You can take a look. And to me, this seemed worth my while. I figured, this is a big deal. I would like to spend some time and see if I can make a contribution to maybe not solve this issue, at least help a bit. So it's a big deal. A lot of people are affected, and the cost is, it's a lot of money that goes into treating or taking care of osteoporotic patients. So a little bit more about that topic. So obviously you know about the human body. Most of you have taken biology. Your body is composed of quite a few bones, and it's actually an organ. Bones it's a living organ. There's a lot of remodeling that goes on. So your bone structure, the way it is right now, might be very different 10 years from now, depending on if you do exercise or not, or you're sitting in a wheelchair. So my research really looked at what is the relationship of the small-scale chemistry. If we look with a microscope at bone, we see all these little pores and little features. What is the relationship of that small-scale structure in chemistry on the macro-scale mechanical competence of bone? In other words, maybe my diet affects the chemistry, or maybe my lack of exercise affects the chemistry, and that in turn makes me more or less likely to fracture my pelvis when I'm older, right? So that's sort of what I did for my PhD. That was the topic that I chose, right? And then to solve that, what was my contribution to the field? Very small contribution. To me, it seems great, right? To me, it seems great, but it was really a very small contribution. I did some engineering, which I probably won't mention in detail. I did some engineering, and I did some applying of the things I figured out to this disease. So the engineering I did was I came up with 2 ways of looking at bone, 2 ways of imaging bone, using infrared imaging. It's essentially a chemical probe, allows you to look at the chemistry. And I did X-ray imaging to look at the structure of bone. And essentially, my PhD involved developing those 2 techniques and then applying those 2 techniques to figure out what the relationship is between taking certain type of osteoporosis medication and its effect on the strength of bone. Now, I wanted to mention this program because I think it's awesome, the Suly Program. Maybe some of you have heard of it, maybe not. So the lab that I did my research in is the Department of Energy National Laboratory. And they have this program where any of you can apply to do an internship, and you get a ton of money. It's usually a summer. It's really, really cool. So check it out. If you type in Suly, S-U-L-Y, in Google, I'm sure it will pop up. So this is kind of like a plug to you. If you're looking for something to do, if you're looking for an internship, you can check this out. They'll house you, they'll give you a stipend, and you basically do cutting-edge research for a period of the summer, 2 months, at one of those labs. And some of it can involve physics or bio or environmental science, depending on what you're interested in. So I've mentored students in this program, I think about 8. And overall, this is really, it's an impressive program. So take a look. Now, just to round it out, the introduction to myself, this picture looks familiar. This is my workbench. Okay, this is where I do my work. And on that little flyer that's out in the hallways and stuff like that, I saw a picture just like this. It's a different one, but this is a synchrotron, and it's a big building. It looks kind of like a particle collider. It has a lot of similarities, but it's a little different. And it's a synchrotron, and it does one thing. A synchrotron does one thing. It makes light. A synchrotron makes light. Very, very basic. Now, it doesn't make light for houses, like light that you can use to, for your light bulbs. It makes light for the purpose of research. It makes light for fancy microscopes, essentially very, very high-powered, high-brightness light with very specific properties. That, in a nutshell, is what a synchrotron is. So I worked at one of these big buildings, big facilities, and most of my talk going forth is going to talk about this. I'll tell you a bit about what is a synchrotron, how does it work, and then I'll talk about some science. I'll have 4 science examples. So maybe this is a review, but I think it's important to put this slide on here. Light, as you might know, can have different wavelengths. You can think of visible light. You've heard of infrared light, but it's a continuum. It's all the way from gamma rays all the way to radio waves. That is the electromagnetic spectrum. So when you think about light, it contains all the, it spans that entire range. Some light that comes from outer space can penetrate the atmosphere, like visible light and radio waves. Some light cannot penetrate because it's blocked by our atmosphere. So some astronomy, we have to put satellites on orbit, things like that, or on high mountaintops because it can't penetrate the atmosphere. And then just for reference, obviously the light has a certain wavelength and they roughly correspond to these objects. So if you're talking about visible light, wavelength is very tiny. If you're talking about a radio wave, they can be hundreds of meters long. One wavelength can be quite long. And light can be used to probe things. So when you do microscopy, you can use the properties of light to learn about the chemical features of your sample. So what I've done for my Ph.D. and for my research now is I've used infrared light, I use X-rays, and those allow me to look essentially at whatever samples I have and learn about the chemical properties, the physical properties, things like that. Right? So a synchrotron produces light. Light contains all those different components, and you have to figure out which component do you want to use for your research. What makes sense? Now, quick quiz. What kind of light is used here? Scanner at the grocery store. Is it infrared light? Infrared light? It's actually just visible light. You see the barcode? That's just visible light. What about that one? That's infrared light. What about that one? Cell tower. Okay. Microwaves. It could be a radio. It could be radio. And last one actually has two. It has 2 answers, but X-rays, you could say terahertz, but X-rays, yeah. So you have interacted with different types of light, on a, maybe not a daily basis, but almost on a daily basis without knowing it probably. So now the unique properties of light, I wanted to highlight this one weird slide that I was like putting it together and it's like, that's kind of neat. I'll keep it in here. So we have a 100-watt light bulb. And let's say I screw it in, turn it on, and I want to figure out that 100 watts, how much does it illuminate per square millimeter? So it turns out if I do the calculation, that 100 watts spread out over that amount of surface, it's going to give me a power density, a brightness of about 0.0001 watts per millimeter.
Incandescent bulbs put out significantly more heat than they do light.
Yes. Yes. You're right. So lasers are more efficient in that sense. They focus all the light on one spot. So it might maybe it's not a 100-watt light source. It might only be like a milliwatt, but all that light is put into a small spot. So the brightness, the density of that power is a bit more than a 100-watt light bulb. Now the synchrotron that I do my research at, it turns out it's only about 30 watts or 40 to 50 watts. It's basically just a light bulb. The difference is it puts all that light, all that power in an extremely small spot, much smaller than a laser beam from a laser pointer, and therefore the brightness is a lot more. So if you can do microscopy, you might have done microscopy in high school where you have a little light bulb in your microscope. This is the grown-up version. It's way, way more powerful. And you can see things that are smaller. You can see things in lower concentrations. Right? So that's why I do my work at a synchrotron and not just in benchtop in a lab anywhere. Now, some of these slides that might be pretty obvious. I'll skip over. So light has, you can have a small light, big light. You can have different brightnesses. These are different properties of light. Now, how does a synchrotron work? You have this thing that spins electrons in a circle. How does it produce light? Turns out you can use magnets to do that. So this is an inside look. This is kind of like that picture on the flyer. I don't know if you saw the top right, similar. So if you go into a synchrotron storage ring, you'll see this. There's about 1,200 magnets. And those magnets are the heart of the synchrotron. They essentially produce light. They produce light spanning from infrared to visible to X-rays. And that light is what we use as a researcher. Okay? So essentially you have like this tiny little pipe over here. It's tiny. It's about, maybe about 1.5 inches. And there's electrons that flow through that in a big circle about half a mile. And the magnets essentially push it around bends each time. That's basically a sort of a circle. And every time that happens, every time a giant magnet pushes those electrons around a circle, you get a little bit of radiation coming off. Imagine driving a car going 60 and you take a quick turn, you'll feel the force. That's my analogy. So you have electrons, they want to go straight, you force them around a bend, and you get some radiation. And that radiation they figured out about 60 years ago, it's actually pretty useful. You can make that turn into a very specific wavelength. So you can produce X-rays or infrared, and that's how you start your scientific project. Another important thing about this light is that light wants to divert. It doesn't always want to go in the same spot. So we do a lot of work. I can spend hours talking about all the work we did to put all that light that this machine produces into a small spot. And essentially, if you have a light bulb, it's going to illuminate everything. We don't want that. We want to put all that light in a very small spot. So we use all kinds of fancy collimators and slits to block out light that we don't want and only collect the light that we want. And what it looks like in a lab setting, now imagine you have your light going from the right side all the way to the left side with all kinds of pieces of equipment that modulate and reshape the light. Take out frequencies we don't want, reshape and collimate. Sometimes you even want to polarize your light. So you can take that light, before you do your experiment, you want to take that light, whether it's infrared or X-rays, and you want to play around with it. You want to tune it as best as you can to match your scientific endeavor. Okay, another map. There's only 4 of these in the US. This big building I showed you, that one over there is New York State. There's another one in Cornell. That's why I did my postdoc there. There's one in Chicago. That's the one that has a picture on that flyer outside. And then there's another one, a quite old one, in Stanford. So if you want to do this type of research, well, you got to travel. There's only 4 in the U.S. And there's there's a few more internationally. But it's certainly it's not easily accessible. There's a process of applying to go to one of these things. It's kind of involved, but that's beyond what I want to talk about. So that's the physics of what I do, right? I don't want to talk about the engineering portion too much, but what is it that I actually do with all that stuff? So as I mentioned, for my PhD, the task was to combine engineering and to combine and some interesting biomedical challenge. I told you about my research in osteoporosis and bone disease. I covered that portion. And more recently, the more recent 7 years, I've taken all that experience that I got in grad school, and I combined that with all the little things I learned about this weird machine, the synchrotron, and I decided to branch out a bit, to not stay within that field of biomedical engineering. Even though I liked it, I had curiosities elsewhere. Right? So that's what I've been doing the past few years. I kind of branched out. This is roughly the science mission. If I had to give myself, what has been my science mission the past few years? This roughly describes it, my science mission. So I elucidate the interactions, the chemistry, and the localization of trace elements, certain chemicals, certain elements in very small quantities, using X-rays, essentially, using light in the form of X-rays. And it has applications in biology, in environmental science, and in geology. I've limited myself to those topics. It's very, very broad. And just to give you, a little preview of what the data looks like that I get. So when I do an experiment, this is the type of stuff I get, right? It's not like a bunch of numbers. It actually looks kind of cool. So here I have a picture of 2 stems of one plant, one wild type and one a GMO version, some genetic mutation. And we're looking at how potassium and manganese travels up the root of that plant. And how come one plant grows very well in poor soil and one plant does not? Right? You can analyze in all kinds of ways, but my approach using the X-rays, using X-ray fluorescence microscopy, is to take a look at that elemental picture, right? So essentially, it is a 3-color false color image, and you can see there's potassium, a lot of potassium in this nodule on the right side in the mutant, and there is none on the wild type. That is information that can easily answer a lot of questions that you might have. Depending on what the question is, you can look at that and say, oh, this is so much information here. There's a question? What's the yellow? The yellow, I think, is zinc. Zinc. I had to check. By the way, if you have questions, feel free. Yeah.
So does that show more the atomic makeup of it?
So this shows you the relative concentrations. So if you see blue over here, that means there's a lot of potassium right here, and there's almost none elsewhere. This is not in any particular unit. It's a lot of work to convert this to concentration. This is just relative concentrations.
You want to develop the habit of saying, hello, my name is Rachel, first and last name, my question is such.
Hi, my name's Rachel, like you said. What's the units after the 500? What is that?
Oh, that, okay, so the picture on the right, this is a tip of a leaf. There's the big leaf over there. That is 1 millimeter, the little white bar, and that is a micrometer. Yeah, so you have a meter, 1,000 times less is a millimeter, 1,000 times less is a micrometer. So meter, millimeter, micrometer. Can't even show it. So the point of that picture is to show that we can take something that, you know, it's the size of your thumb and look at it at fairly high magnification. And then, depending on what your question is, you might be able to answer, oh, there's a lot of arsenic on the tip of the root. How come I don't see arsenic anywhere else? How come I see calcium concentrated in little weird little fibers here? Right? A picture like this often tells 1,000 words. I'll have more examples like this. I just wanted to preview what the data looks like. Another example, closer to those of you who are in biology, is what's a hot topic right now is to look at the interaction of roots of plants via their root system with the soil. You can't just take a plant and put it in a different soil and expect it to grow. You can't just put it in a different soil and give it whatever nutrients it needs. It needs a rich, it needs basically a collaboration. The plant collaborates with fungi to make all those nutrients bioavailable. And those are questions that up to a few years ago were very hard to tackle, but the lab I work with has developed some tools to do that, right? So we can take a root, section it, and essentially look at a little root structure, and then look at what's called the rhizosphere. Look at the small area around the root where there might be fungus, and figure out the interaction between the fungus and the root. Turns out it's a beautiful collaboration. The plant gives something to the fungus, and the fungus gives something to the plant. There might be nutrients that the plant needs, but the plant can't get them because the nutrients are locked up in a certain chemical formation. And maybe the fungus can unlock that. The fungus can make that bioavailable to the plant. So it's a cool relationship that we can now study with all these new tools. This is one of them. So that's an example in biology. Now, if you're wondering, like, what could I do with this synchrotron thing? It's often a conversation you have. When I talk to someone, it's a conversation I have with a potential researcher. I learn a bit about what they do, what questions they're facing. I tell them things like this. Here's what I do. Let's see if I can help out. So some things to consider, if you're wondering. One of the hard things is, how do I get a sample from your lab to my lab? Often it's not easy. Maybe you're working with soils. Maybe you're working with rocks. Maybe you're working with biological specimens, like human tissues or animal tissues. And the hard part is often, how do you properly prepare those samples so they're still intact, they're still giving you information, at the same time I can analyze them. It's not tricky. How do you mount them? Do you cut them? Do you freeze them? Do you keep them dry? Do you hydrate them? All those questions. And the next thing is we go to the beamline, as it's called. The microscope's called a beamline. And we use X-rays to excite trace elements, and then we get cool pictures like this. False color image, where we're looking at maybe the calcium content in the leaf. Very high quality, high resolution images, and hopefully that is part of, it can solve some of your questions. We can look at different elements at the same time, so we can look at calcium and zinc and magnesium and iron and sulfur and arsenic all at the same time. So you get a rich amount of information. So often, you're dealing with the problem. Getting the data might not be the hardest part. The hardest part might be going home and analyzing that stuff. So that might be the hardest part. And why I sort of mention this, like what is roughly the scientist that comes over and uses this technique? Well, it's people that explore the interaction chemistry and localization of trace elements in whatever field they're in. And some examples include cleanup of contaminants, environmental science. Maybe there's some people from agriculture that look at crops. So those are some examples of people that I often work with that come over and collaborate with me on this type of science. I'm going to skip that slide. So I have one slide on the engineering portion. It's hard to capture the microscope that does all this stuff. But essentially, we built one from scratch. So it took us about 3 to 4 years to build one that can do all that cool stuff. So you have that big building, the synchrotron, and then that produces light. But you still have to make the microscope that takes that light and does something cool with it. And that's called a beamline. And essentially, we built one in about 3 to 4 years. And these things are oddly shaped. There's quite long, all kinds of wires and things like that. So my best, this is my best shot at giving you a view of that. So that's me over there standing on top of one of the hutches, looking at one of the ends. You can see a lot of pipe over here. There's a huge pipe. And essentially, that is where the light goes through. This picture over here is simply a zoom in on this picture. Whatever sample you have, your sample goes right there. Right? So you can't look at big things. You have to look at small little things and prepare them in a certain way. You stick them right there, and then we can analyze them. We have several detectors that detect whatever signal you're looking for. So it could be an X-ray fluorescence detector or an X-ray diffraction panel. We obviously have a little visible light image so you can see your sample and correlate it with visible features. You can do all kinds of things. You can come in with your own equipment. So I had some people that did high pressure work, essentially 2 big diamonds, actually 2 small diamonds, that when squeezed together produce a very high pressure. And maybe if you're in geology, you're interested in looking at certain chemical reactions under very high pressure. So you can bring in your own cool devices. It's a very modular approach. You just pretty much just come up with a cool device. You can pop them in there and then adapt this entire microscope to suit your needs. So it's quite versatile. Let's see. OK, so that concluded the introduction to me and the introduction to my field. Next, I have 4 science examples. But before I go to that, is there any questions on any of that stuff? Yes?
Hi, my name's Tabitha. I was going to ask you, do the samples you have, if you're using X-rays for them, does the radiation cause any issues? Or?
Yes, it does. That's a very good question, yeah. Yeah, so imagine an old newspaper. They'll be photo bleached. The same thing happens with our samples. Some samples are more resistant to it. If you're looking at geological samples, often they'll be fine. But if you're looking at very delicate samples, like we do some cell culture work, looking at individual cells that are in a hydrated state, looking at them with X-rays is going to burn a hole in them almost. So it can be very tricky. And in some instances, like looking at biological specimens, you have to take precautions. And one of them can be freezing your cell, with liquid nitrogen, or you can substituting water with glycerin, things like that. So there's a big concern. Yeah.
Hi, my name's Morgan, and I was wondering, like, every result you get, is it 100% accurate? Because of the time?
Okay. Yeah.
Is there anything that can interfere with it, and because if the sample gets ruined and you don't have another one?
Right. Right. So I'm showing you success stories. Yes, and there's a lot of failures, for sure. And that's why, as a researcher, you work with what's called a beamline scientist. You work with someone like me. I'm the one that sort of validates the data you get. And if I say, to my knowledge, this is good data, then that's your best guide. So we do a lot of work. We spend a majority of our work verifying our techniques, always comparing it to a gold standard, a different technique. If our technique gives the same result as a different technique, then we have some confidence that what we're doing is producing correct results. But a lot of work goes into, there's a lot of data that we get. It's like, is this real or is it not? And that's a concern. That's a big concern. And depending on the science you're in, sometimes you want to collect more data. You want to do an experiment over and over and over again. If you can get the same exact result in my lab and in one of the other labs I mentioned in Stanford, if you can get the same result in 2 labs, you're probably on the right track. Right. So it's a big concern. Yes, it's a big concern. Oh, back there.
I'm Melissa, and our topic tonight. Kind of going off our question with the radioactivity, the synchtrons, does it pose a health risk to like people itself, not just the specimens or the project, like the surrounding areas or anything like that?
Yes, it does. It does. So what I didn't include is that the experiments that we do, in other words, when you have a sample and you are ready to analyze it, you put it there, you mount it on your sample holder, then you walk out of this room, you close the door. It's basically a lead enclosure. And then you turn the machine on, basically in a nutshell. Yes, it's a high radiation environment. So humans are not allowed in that little lab space. Yeah, it's a big deal. Yeah, we take that very seriously. Everyone wears dosimeters. You get an annual report. It should say 0. If it doesn't say 0, a lot of bad things, a lot of administrative things happen.
Hi, my name is Caitlin. I was wondering if you or the other people that work there, if you ever have to get like radiation checks just to make sure that everything's working and you're all right.
Well, we wear dosimeters. And a dosimeter is a little device size of a credit card, and it continually measures the radioactivity that you're exposed to. And at the end of the month, you turn it in, and they give you a dose. And it should say 0. If it doesn't say 0, we have to figure out where were you? How come you got a dose? You shouldn't get a dose. So you can't insulate yourself. You can't protect yourself other than walking away from where the radiation is. In other words, stepping out of that little room where your experiment is taking place. And the administrative control, there's several administrative controls, but part of that is wearing a dosimeter. So, yeah. The radiation that you're exposed, or that you would be exposed to, it essentially it goes through paper and it goes through clothes. So you have to be cautious of that. Yeah.
Dr. Acerbo, my name is Sarah. I'm curious, you were talking about electrons being pushed around. These are like atoms, electrons?
Yeah. Electrons. Yeah, it's an electron storage ring, and they're basically, they're...
Where do you get those electrons from?
Oh, okay. Well, that's yes.
Okay,
so
sorry.
Yeah. That's a good point. There's an electron gun. It's a device that essentially produces the electrons. It shoots them in a straight trajectory, and then they're accelerated with fancy devices, radio frequency cavities. They're accelerated to 99.999999% the speed of light, and at that speed they shoot around that giant building. That's the electron storage ring, and then there's about 50 or 60 microscopes all along that ring. Whenever there's a bend, we capture stray radiation and we produce different types of light. So, we use electrons, we generate them in very specific ways, and we keep them in that orbit with a lot of magnets. That's not the part that I do. That's a whole different side.
Can I continue further and ask, those electrons, do they have a lifetime window? Meaning, can they go only a certain number of times around before they...
Yeah. Oh, yeah, totally. So just keep an eye on my time over here. Yes, that's a big concern. So the amount of electrons in that storing ring decays. So you constantly have to add more. Yes. Yes. The short answer is yes. We could talk more if you're interested. So I want to talk about some science, and I'm going a little over time. So I'm gonna, if there's any more questions, let's save them for afterwards. Okay. So this is my first science example. This is environmental science, fracking. You've heard of it? Yes. So now I am not an expert in fracking. I'm the person that works with people that do this science. So I'm the guy that helps do the experiments at the microscope to make these things possible. So I know just enough to make these experiments successful, right? And I'm going to give you a little background and then talk a little about how I come in this project. So fracking, as you know, can be used to obtain trapped gas from underground. And one of the major problems is that it produces a lot of pollution in the form of solid waste has to come up. And what do you do with that waste? It's not regulated. And more so than that, this process of fracking, it's essentially you take a lot of water, you add a lot of different solvents, you push it down a big tube, and then you get gas in return, but all that water you pumped in there comes back up. So you have what's called waste water, produces wastewater. And that wastewater is a big deal. It's a lot. Where does the wastewater go? It's got to go somewhere. So it's going to go to the water treatment plant nearby. And yeah, basically, the wastewater's got to go somewhere. It's basically going to go to the water treatment plant. And if your water treatment plant is not set up for that, then you have a problem.
Exactly. That's
yeah, and so that's been one of the problems, been one of the concerns. So if you take water samples from a domestic wastewater, non-fracking wastewater, you can quantify it, the total dissolved solids and certain ions, right? And it turns out that in fracking wastewater, you have a lot more of that. The concentrations of those are way more. And water treatment plants might not be set up to properly take care of that. Okay, so that's sort of the problem. Here's a science problem. Let's see what we can do. So geographically, here's what we're focusing on in this research project. It's a huge deposit roughly spanning Pennsylvania. And here you can see just a map showing the extent of that deposit and the depth in feet. And I have a similar map. In this case, this is showing if I were to take a sample of wastewater in each of those counties, how much of that bad stuff is in there, how much of total dissolved solids, how much of certain ions are present in there, and darker means more. Pretty much tracks the picture I had on the previous page, right? Where there's a lot of shale, there's a lot of wells, and therefore the wastewater goes into the public water supply, and there's problems in that sense. And this basically shows you the extent of all those wells where all that fracking occurs. Perfectly follows, of course, the presence of shale. So the problem is you get all this high salinity wastewater. How do you treat that in a water treatment facility? So the problem with that is you can't do bacterial treatment because the high salinity kills that. It's corrosive. It disrupts commonly used treatments to take care of very trace quantity of certain elements. And because of all that bad stuff, there's compound effects that we want to look at in this particular science project. What we figured is, if all that bad stuff happens, I wonder what else is going to happen. So it turns out we're going to look at precipitation of barite, certain type of compound, and we're going to figure out if that barite, if it sits there long enough, if it can absorb other bad stuff, particular radioactive materials that are buried on the and if that can essentially get into a water supply. So it's bad stuff. Let's see what we can do here. So I worked with a scientist. That was a problem. And essentially I told scientists, well, here's what we can do. We can look at really small stuff. So if you give me a water sample that has some of those precipitants in it, maybe we can figure out what is the chemical makeup of those precipitants. And maybe we can figure out what does the evolution look like of those little solids. So this is essentially looking at that type of, trying to answer that type of question. So we have those tiny little particles that form in wastewaters, and we want to know those tiny little particles, as they grow bigger and bigger and bigger, do they incorporate all those bad things, all those things you don't want in your public water supply? And some of the pictures that we can produce, we can look at single particles. These are very small particles, 2 microns. And with X-rays, with X-ray imaging and X-ray spectroscopy, essentially we can answer some of those questions. So we can look at, okay, here's a particle. Let's look at the barium distribution. So this image is showing a fairly homogeneous barium distribution. If we look at arsenic, something you don't want, it turns out arsenic gets trapped in those barium particles, and there's a very particular zonation that occurs. And that essentially, in this project, that caught our interest, and we decided to do quite a bit more work on the evolution of those particles. Maybe there's a way, maybe there's something very simple we can add to prevent that process from happening, to prevent those barium particles from taking up all the bad stuff. Maybe we can saturate them with stuff that we can easily treat, that's not as dangerous, and we can prevent those barium particles from absorbing the really bad stuff. Right? So this is an example of an environmental science project. Gave you a quick introduction, and this is essentially how a microscope like the one I work with can answer that question. Now, if you enter chemistry, you can take it one step further. You can say, I got a particle, and I can see exactly where the arsenic is. I want to know specifically what oxidation state that arsenic is in, and that can give me other information. And you can do that too. You can do that with X-ray spectroscopy, essentially. And with X-ray spectroscopy, you're essentially taking light and you're changing the wavelength a little bit, and you get what's called a spectrum. And that spectrum can tell you a lot of detailed information about what oxidation state of arsenic you have, or whatever element you have. The next example I want to give, second example, is more along the lines of agriculture, crop science. So in this case, we're talking about rice paddies and growth of rice and the prevalence of arsenic. So some of you may know arsenic is naturally present in nature. So it's a toxin, but it's naturally present from all kinds of different events. So if you make a map of the US and you roughly sketch the prevalence of arsenic in the ground, you will get something like that. And it turns out this Mississippi Delta over here, that's where we grow a lot of rice. And rice is really good at taking up arsenic from the soil and putting it into the rice grains. That's why if you look on the FDA, it says no more than 2 servings of rice a week, because you have elevated arsenic in your blood, in your body. So don't go eating 50 rice cakes. Might not be a good idea. So that's the problem at hand. So the problem at hand is we have a lot of arsenic in the soil, and we grow rice. Some forms of rice are really good at taking the arsenic. It's called a hyperaccumulator. They accumulate the arsenic way up into their little rice grain, and then we can eat that. Okay? So that's the bad thing. And we want to figure out, is there something we can do with the way we grow rice? Maybe we can come up with a different strain of rice plant, or maybe we can do something with the soil so that this doesn't happen as much, so that the rice doesn't take up as much arsenic. Right? So this is more of a agriculture, crop science, geology type of project. And it turns out that it has a lot to do with the chemistry of the soil, and mainly it's directed by redox conditions and by microbial activity that make arsenic in the soil available to the plant. I mentioned that about 20 minutes ago. A plant might want what's in the soil, but it might not be able to get it. It can only get it if the conditions are right, the chemistry is right, and if the microbes are there to make all that stuff available to the plant, right? So maybe we can play with the redox conditions in such a way that the arsenic is not as bioavailable to the plant. So I'll spare you some of the details on this one, but essentially what we're doing here is we're playing around with alternating wet and dry cycles on rice paddies. Instead of having a rice paddy where it's always wet, maybe we'll take a look at what happens if you flood it and then you dry it and flood it and dry it. There are certain strains of rice that are totally fine with that, and it can change the microbial activity entirely. It can change the chemistry entirely. So let's take a look and see if that does anything. It turns out it does. It turns out you can a nice side benefit is you're reducing water consumption, but also in some cases you're reducing arsenic uptake by quite a bit in those plants. That's a good thing. So here's some pictures from the field. I wasn't there, but I thought it was cool for you to see. That's where the science starts, like literally in the field. Let's dig some holes, and the science ends with cool pictures like this. These are cool pictures of tiny little pieces of soil. So someone literally took a shovel, put it in a little bucket, and essentially sent it to me. And I went through a sample prep, and I took pictures like this one. This is a tiny little grain of sand. It might seem insignificant, but it tells you a lot of information. It tells you a lot about where, in blue, is manganese. It's a very relevant element in this case. Where is the iron? But more importantly, where's the arsenic? We know there's arsenic somewhere. How does that, is it co-localized with iron or with manganese? So this is the type of data we can get with a microprobe, the microscope that I built. And then you can make plots like this, and you can say, aha, looks like the arsenic and the iron, or the arsenic and the iron are co-localized. So there's something going on between arsenic and iron, and the manganese is sort of an outlier. Right? So we can get elemental and elemental localization of these elements. And of course, we can go a step further. We can go a step further and do some more detailed chemical analysis. We have arsenic, great. What form is it in? We can do spectroscopy to learn a lot more about what type of arsenic do we have. Arsenic 3, arsenic 4, where does it come from? Maybe we can figure out where it comes from. And that's when you get spectroscopy information like this. This looks like, I don't know, a star system, a nebula or something like that. This is actually some soil. And the exciting thing to the researchers was, what are those little red dots over there? How come there's those tiny little red dots that are only arsenic? And it turns out that had a huge significance. And we were able to do, let's see, my pointer's a little lagged. Turns out we can even focus in on those tiny little red dots and do some detailed spectroscopy and learn very detailed information about those micron-sized little particles to kind of round out the science project. Okay, I got time for one more project. This one deals with actually something that is sort of relevant to Vermont, I think, as well, or hopefully not. This has to do with the Hanford nuclear site in Washington State. And what happens here is they have a lot of nuclear waste that's buried. And they did that a while ago, and I figured we'll just sort of try to contain it as best we can. And then over time, as we get more technology, we'll figure out how to remediate this, how to deal with this waste. But in the meantime, all that waste that's buried there in that blocked off site over there, it's leaching and it's causing problems. So there's a lot of money going into figuring out how can we contain that, how can we prevent all that nuclear waste from seeping into the water supply, and maybe even how can we properly get rid of it. Right? So some of the research I did involved that. So the picture is nuclear, is Hanford nuclear site, and here's the problem. You have waste, then you have some type of layering here, but in the end of the day, you have groundwater, and that groundwater is going to seep up into where the bad stuff is, and it's going to basically leach it out. So you get, you essentially get a uranium plume into your groundwater. So how do you prevent that from happening? Okay, so here's the problem, and let's figure out what we can do. Let's figure out maybe how we can make a contribution to this field. So this particular science, we had a different approach. We said, let's try to model what's going on in Hanford in the laboratory. So we have this, in the Hanford nucleus site, you have a lot of soil and different soil composition, different porosity, and you have weathering effects, and you have your contaminants. Let's try to recreate that in the lab on the small scale. So that's what we tried to do. So we came up with this weird little device. It's basically a microfluidic device, and we decided we're going to pack in all the chemistry into that microfluidic device to mimic what's going on in the real condition, and hopefully we can then investigate that chemistry at our beamline. That's what we decided to do. So there's all kinds of engineering challenges that go along with that. How do you mimic weathering? How do you mimic the flow of chemicals? How do you still observe what you want to observe? Right? So there's all kinds of material science tricks that you have to play. But essentially, we sort of solved all those things, and you can see our little device over here. It's like a little square thing, and we can flow in liquids, and we can flow them out to mimic weathering, to mimic like period of rainfall or whatever it is. And then once we've done our experiment, our data essentially would tell us exactly how certain elements might flow around certain cavities. So if we have, for example, arsenic, how does arsenic interact with the ground in different porosities? Does the arsenic mix with some of the other constituents in there? So this is information we can get by keeping the sample intact. We're taking our sample, we're keeping it intact, we're trying to mimic whatever's going on in the ground, and we're trying to answer some of the basic questions in the lab. So we can take cool images like this, and then we can also do spectroscopy to learn a lot more about the oxidation states, and that might give us a lot more detailed information about the problem at hand. I'm going to skip that one, and I'm going to go to my summary slide. So I think I'm basically almost there. So what I want the take-home message to be, I have two summary slides. The take-home message for you today is a degree doesn't necessarily lock you into that field. So my degree was biochemistry, computer science. And as you see, the research I do, it spans other stuff as well. I did physics, mechanical engineering. I did some environmental science, geology. I worked with some samples from the moon from Apollo missions, things like that. Yeah. And the microscope that we built, who knows, maybe it'll be used for Mars return samples, right? You can think of those things. So as someone with a degree in biochemistry and computer science, I'm doing all these other cool stuff, all these other cool things. So the first thing, you get a degree, don't freak out. You can still branch out. I think interdisciplinary sciences are gaining popularity. You can be a chemist and make that your career. You can be a biologist or a doctor, you make a career. But if you are the type of person that likes to combine two sciences, totally cool. You can do that. There's a lot of need for those people as well. Someone that can do chemistry, but is also good with physics or with computer engineering. Another thing is branch out, meet people. That's how you get ideas. That's how you discover what's out there. Coming to things like this, coming to topic, to speakers like this. Now, this is, someone that I know mentioned this. Find out what you like and what you're good at. If you can find those two things, you probably have a good time. So, for me, I was good at chemistry and I liked it. I was good at computer science, I liked it. I stuck with it. Thumbs up. And then just a summary of the science. I did not mention the last one, but the first three I spoke about trace elemental composition in those little Barry particles, the wastewater fracking. And that was roughly environmental science and Department of Energy style type research. We spoke about the rice paddies, arsenic, and how it is affected by alternating wet and dry cycles. So that's a biology and agriculture, crop science type of experiment. I spoke about pore scale processes in that nuclear waste site in Hanford. And for that, we did some engineering, and we looked at environmental science type research, and it was a little bit of biology as well. So those are the three topics that I covered. And with that, thank you. And these are the collaborators I worked with. So here's some of the students I worked with over the past years and some of my collaborators. There's too many to put on there, but just a few. So with that, please, any questions?
Thanks, Sarah. After last week, I think all of us would like to say thank you for taking the time to make it lay people friendly and as well as to speak slow enough that we could all take notes.
Oh, good, okay.
It's very appreciated.
Awesome, thank you.
Thank
All right.
you.
Your name?
I'm Alex.
Alex,
And
yeah.
you mentioned something in the title that the radionuclides in the fracking.
Yeah.
If you could just elaborate on what those are.
Well, there are certain radioactive materials that are buried down deep that are normally not a concern. They're way down there and they're out of sight, out of mind. But if we're fracking and we're disturbing the geology back, underground, they can be dissolved. They can come up with the wastewater. So that's the problem. So things that are normally not a concern, now they can come to the surface by means of fracking. So it's a bad side effect of fracking. And the question becomes, can we prevent that? And if not, how can we remediate that? How can we take those out of the water that comes back up, that wastewater? Yeah.
And then actually also in the same little vein, you mentioned a PPTE treatment for removing wastewater in that same analysis.
Yeah.
Oh, okay. So what I meant with that is when wastewater is treated, they have a spectrum of chemicals, and they want them down below federal standards. And some of them are in the PPT range. Now, if you have water that's loaded with all those contaminants, it might be very hard to apply your commonly used treatment procedures to get your water within regulatory limits. That's what I was referring to. Yeah. If your water is loaded with a certain chemical, it might prevent you from reducing things down low enough. Yep. On this side, in the back. Oh, back there, yeah.
Has this methodology been applied to cancer cell biology, specifically looking at differences in tumor cells versus normal cells?
Yes. So the question was, has this type of have these techniques been applied to cell culture work and specifically cancerous cells? Yes, totally. I referenced, I forget your name, but you asked a question about radiation damage. It's a big concern. Doing cell culture work is totally possible. There's a lot of people who do that. You can look, you can interrogate cells. You can envision doing antibody staining, tagging certain components, and having the tag be a fluorescent marker, like an osmium, a big signal. You can image that with this type of stuff. The problem is the cell damage. In terms of everything is done. Everything is done you have to work with cell cultures, right? So that's the trick. So if you can model your system with cell cultures, you have a chance of doing it here. The problem still is often that you have radiation damage effects, and they can obfuscate your results. Yeah, I did a little bit of work with cancer cell work with infrared imaging, which is probably more appropriate, not so much X-ray imaging, because infrared imaging gives you direct chemical information. X-ray gives you elemental speciation, which might not be as interesting if you're doing cancer cell research. So there are quite a few people that do infrared work, but it's tricky. The radiation effects are problematic. Yeah. I saw someone else in the back. Yep.
Melissa, I just am curious, when you decided to do the osteoporosis, was it just because it was just so interesting, or did you actually go take a look or somewhere from it?
Well, that's a good question. Why did I pick osteoporosis? Well, the grad school that I joined, Stony Brook University, has at the time had the number one, osteoporosis research group. So I joined that group, and I was surrounded by experts, and so that made it fairly easy to stay in that field. I had all the tools I needed to dig right in. So that was probably the main reason I stayed in that line of research for my PhD. Yeah. And then once I got my PhD, I sort of branched out. I did some structural biology, looking at protein structure after my PhD, and then it snowballed from there to what you saw in my presentation. So, but as a grad student, I mean, often you have sometimes you're sort of given a topic, but you have still a lot of wiggle room. Well, I want to approach from this side, or I want to include that, right? You have a lot of creativity if you're trying to pick your, if you're picking a project for your thesis, you have a lot of flexibility in what you want to do. Yeah. Okay.
Thank you very much for your introduction to synchrotron radiation.